Hi class, how are you? Today I will be starting chapter 2 of this course. And the chapter 2 is cell structures and cell division. And so what I've done is I've divided this class into two parts. Number one, in one section, I'll be talking about cellular structures and about lipids, and then about mitochondria. So three topics in one section, and in the second section, I'll be mainly talking about cell division, and and in this first topic will be mitosis, and this third will be meiosis. So altogether, there will be six topics, but I will be uploading two videos. First video for cellular structures, lipids, and mitochondria, and the second video for cell division, which would include mitosis and meiosis. So let's get started, and let's look at the learning objective for this chapter. So the learning objective is to compare and contrast prokaryotes and eukaryotes, know the function of basic organelles. So what are organelles? Organelles are those small units within a cell that helps the cell to function, and that we will see. And then know the physiochemical properties of cell membrane, which essentially means looking at detailed picture of a lipid molecule, enzyme catalysis, and then the second part would be DNA polymerase, mitosis, and meiosis, as I discussed before. So let's, before I go into this chapter in detail, I would like to give some basic concept. What is the significance of this chapter? So, we can divide this whole planet. I'm just talking for the sake of increasing our understanding, into two parts. Part one would be living things, and part two could be non-living. So let's say what could be the biggest property of a living thing. So the major properties of living thing would be, number one, to grow, to develop, and differentiate. These are the three properties of a living being that is not present in any non-living material. So, so what is the fundamental unit of life? A fundamental unit of life is a cell. Why it is called as fundamental unit? Because it can survive on its on its own, a cell. Now, what we know that biology is a very big, very diverse biology or biological organism or living organism. Organisms are very diverse, huge. Why I say huge? Because it ranges from bacteria to plants, animals, and this also includes human beings. Huge catalog of living organisms. So when we want to study, what is the best way to study this diverse organisms? Is to organize it, is to classify it. So scientists or biologists, they have divided the living organisms in many ways. For example, it could be all the living organisms could be divided on the basis of, let's say, number of cells. It could be divided on the basis of number of cells. And based upon this, there can be two types. One, a single cell organisms, and second, multicellular organisms. Now, in the single cell organism, the example would be bacterial cell, or algae, and let's say fungus. They could be single cell. Now, in fact, all of these could be, fungus could also be multicellular. And the second would be multicellular, such as plants, such as human beings. So this is one way by which we can organize living organism, biological organism. Second way we can organize is through, let's say, presence of chloroplast. If chloroplast is present, then these are plants. If chloroplast is absent, then they would be part of animal world. So first, it could be depending upon the number of cells. Second, by presence or absence of chloroplast, defining two kingdoms, plant kingdom and animal kingdom. The third way, this could, again, organisms could be divided is on the basis of presence or absence of nucleus. So in organisms where nucleus is present, it could be referred as eukaryotes. And in organisms where nucleus is absent, prokaryotes. So eukaryotes and prokaryotes. So let's revise. We can distinguish organisms based on the basis of number of cells between unicellular and multicellular, on the basis of chloroplast into animal kingdom, plant kingdom, and on the basis of presence or absence of nucleus, eukaryotes and prokaryotes. So what are the examples of eukaryotes? Such as human cells, plant cells, and an example of prokaryotes will be bacterial cells. So going forward, we are going to look first into prokaryotes. Then we would look into human cells, an example of a human cell, and then we could look into plant cell. So let's look at a bacterial cell. So a bacterial cell, essentially, it is composed of boundary, and this boundary could be protective in nature, and this boundary is referred as, this outer boundary is referred as cell wall. Now within the cell wall, there is another boundary that is present within a bacterial cell. This boundary is called as cell membrane. Cell membrane. Now, so cell wall and cell membrane are two boundary lines that divides cell from the outer environment. And they are protective in nature, they are selective in nature. And just like countries have boundaries, they have border to protect citizens. That's how cell wall and cell membrane helps the bacteria to survive in. Because bacteria is an open environment, they need to have extra protection, they need to survive. Now within this cell wall and cell membrane, there is a fluid structure. This fluid structure is called as cytosol. Some books will also refer this as cytoplasm, but they essentially mean the same. So internal to cell membrane is a fluidic medium, which is called as cytosol, and 70% of this cytosol is water. Now within this, there are, just like a school has various departments like human resources, academics, engineering, library that supports an institution. That's how there are various organelles are present within a cell to support its survival. So, but to note worthy, if you remember what I mentioned in the beginning, is that bacteria, they are prokaryotes. And the definition of prokaryotes is that they have no nucleus. No nucleus is present in a prokaryote, in a bacterial cell. So how is the DNA present? So DNA within a bacteria is present directly in the cytoplasm. There is no nucleus. So this DNA, which is present in bacteria without nucleus, is called as nucleoid. Now, And this nucleotide is essentially DNA or bacterial chromosomes. So DNA or bacterial chromosome. Since there is no nucleus present, so this is called as nucleoid. Now, one of the essential aspect to support bacterial cell is another organelle which is present inside the cytosol of the bacteria. This is called as, this essential organelle is called as ribosomes. Now this ribosome is the main organelle that manufactures protein. So the primary function of ribosome is protein synthesis. So this essentially is the bacterial cell. Now we all know that bacteria needs to survive in an external environment, which could be harsh, which could be favorable. So bacteria, to support itself, they have some extracellular hair-like structures. And these can be of three types. One is long, like a whip-shaped structure, long. And this long whip-shaped structure is called as flagella, which helps in the movement of bacteria, movement or locomotion. And why does a bacteria needs to move? It needs to move towards food. It needs to run away from enemy. So thin whip-like structure is called as flagella. The second type of structure that bacteria has, they are very small and they're present all over the bacterial cell wall. Very small hair-like structure. Is present all over the cell, bacterial cell. Small hair-like, not like flagella. These small hair-like structure is called as fimbria. And what is the role of this? It's called as for attachment. The bacteria is swimming in the water or in the air. They need to attach to food material, or in the water, if the current is very high, it is floody situation, then they need to attach to a rock or attach to a food particle. That is achieved through fimbria. So flagella, fimbria. The third particle the third appendages that bacteria has, and this is called as, this organelle is, this appendage is called as villi. And this helps in the sexual reproduction. Bacteria mainly replicates or divides by asexual means, but rarely bacteria also undergo sexual conjugation. There is no apparent male and female bacteria, but not well-defined sexual organelles, but bacteria that have pili, they are designated as male, and bacteria that don't have pili, they are considered as female. So this pili helps in sexual conjugation and helps in injecting the DNA into the female bacteria. So let's recapitulate very quickly. A bacterial cell is mainly without nucleus. The chromosome is in the cytoplasm called as nucleoid. And one of the most important organelle in bacterial cell is ribosomes that helps in protein synthesis. It helps in protein synthesis. Then cell wall and cell membrane. Cell wall helps in protection. And cell wall is generally, cell wall is dead material. And cell membrane is living, selectively permeable. And 3 appendages, flagella, fimbria, and pili. Flagella for movement, locomotion, fimbria for attachment, and pili for sexual conjugation. So this is about the bacterial cells. Now let's talk about a eukaryotic cell, and the example is human cell. So let's draw a typical human cell. Typical human cell. And unlike bacteria that has a cell wall, human cells, they don't have cell wall. Instead, they have cell membrane. Cell membrane that covers the human cell. So, cell membrane. Human cells, they don't have cell wall. Just to remind you. Now, as mentioned, human cells, they have nucleus. This nucleus is present inside the cytoplasm. Right? So let me, I'll just make again. So this is cell membrane. Within the cell membrane, there is another organelle. This organelle is called as nucleus. Right? And the space between nucleus. So nucleus is housed within a human cell, and this is again called as cytoplasm or cytosol. Now, human cells are very specialized as compared to the bacterial cell. So first of all, within the nucleus of the chromosomes are present the DNA material, but DNA is not present as by itself in the like bacteria, but DNA in human cells are present as chromosomes. DNA is present as chromosomes. And so DNA is present as chromosomes. And if you take human cells, there are 46 chromosomes in a human cell, but in actual it is 23 pairs of chromosomes, not 46 chromosomes. So a human cell or an eukaryotic cell will have cell membrane, and within the cell membrane will be cytoplasm, and within the cytoplasm is the nucleus, which houses the chromosome. So the membrane that separates nucleus and the cytoplasm is called as, some books will say nuclear envelope, but mostly will say nuclear membrane. Now, these are major part of human cell, but they're again supported by small organelles which play a vital role. So let's list them. So one is ribosome, and ribosome, just like bacteria, ribosomes here also, they are important for manufacturing of proteins, ribosomes. The second organelle that is present is called as ER. Now this ER can be of two types. One is rough ER and the second is smooth ER. So ER stands for endoplasmic reticulum. And this could be of two types. One is rough and second is smooth. The rough ER is responsible for protein synthesis, and the smooth ER is responsible for lipid biosynthesis. So two kinds of ER. ER stands for endoplasmic reticulum. The third, the other organelle that is present is called as Golgi apparatus. And this Golgi apparatus, it helps in the protein secretion. It helps in protein secretion. So let's revise. So a human cell will have cell membrane. Within the cell membrane, it will be cytosol. Within the cytosol, there will be ribosomes, primary protein synthesis organelles, endoplasmic reticulum that could be of two types, smooth and rough. Rough is responsible for protein synthesis. Smooth is involved in lipid biosynthesis. The nucleus is separated by the cytosol with nuclear membrane, and within the nucleus are present chromosomes. Now, one of the most important, all these organelles are important, and another key organelles is called as mitochondria. And mitochondria is called as powerhouse of the cell. Mitochondria is referred as powerhouse of the cell because this generates ATP. Now, one thing very important I would like to mention here, there are two kinds of eukaryotes: single cell, like yeast, fungus, or algae, or multicellular, like human cells. So these single cells, they have additional organelles called as lysosomes. They contain enzymes that helps in the digestion. Lysosomes are small sac-like organelles that contains digestive enzymes that helps to digest. And also these single cell eukaryotes, they have to survive, they have to maintain hydration, or throw waste material out of their body or of the cell after digestion. These are referred as vacuoles. Vacuoles, which are sac-like organelles, helps in throwing out the waste material. They also sometimes contain water to help the single cell eukaryote survive under high temperature. So it helps in the hydration, maintaining osmotic pressure within the cell. So what we discussed here, a bacterial cell and a human cell. Bacterial cell represents prokaryotes and a human cell represents eukaryote. Now, very briefly, I will talk about plant cells. Very briefly, I will talk about plant cells, because plant cells are also eukaryotes. But what is the major difference in the plant cell is that the plant cells, they look green. The leaves, they look green. Right? This greenish look to plants are given by an organelle called as chloroplasts, which is not present in animal cells or human cells. And within this chloroplast are present a special pigment called as chlorophylls. This chlorophylls traps solar energy. To remember, plants are primary producers. They can trap solar energy to make food. And what is that food? Carbohydrate, glucose, that helps the plant to survive. We don't have that capacity because we don't carry chloroplast, we don't have chlorophyll. So we have to depend upon. So we are secondary consumers. Plants are primary consumers. So let's recapitulate everything. Bacterial cells without nucleus, they have cell wall, they have cell membrane, they have appendages such as flagella, fimbria, and pili. A human cell is specialized. They have nucleus, well-formed chromosomes. They have mitochondria. They have endoplasmic reticulum, smooth and rough, Golgi apparatus. So they are much more specialized. Plant cells are unique because plants are a primary producer. They have chloroplast, which contains chlorophyll that harnesses solar energy to prepare food. So collectively, this is the basic organization of plants and animal cells, as well as the bacterial cells. So let's go back to the slide. So let's look at the bacterial cell, I'm sorry. So a bacterial cell has a cell wall, the outer membrane. They have flagella, the whip-like structure that helps the bacteria to swim towards food or away from enemy. Ribosomes that helps to make proteins. Then bacterial DNA is present as chromosomes without nucleus, called as nucleoid, and they have cell wall and cell membrane. And fimbria, they help in attachment. So very simple cellular organization of bacteria. If we compare a eukaryotic cells, they have a nucleus which contains chromosomes, and nucleus is separated from other part of the cell by nuclear membrane or nuclear envelope. Within the cytoplasm, you have ribosomes, you have Golgi apparatus, then mitochondria. These are eukaryotic cells, like algae cells. So they contain flagella because they also live in water or environment. They need to swim. Lysosomes. And then you have a rough endoplasmic reticulum, protein synthesis. Smooth endoplasmic reticulum is a lipid biosynthesis. And finally, Golgi apparatus, which is involved in protein secretion. So here I would like to add, so let's say you get a question which says that what is the primary organelles that helps in protein synthesis? The primary organelle is always going to be ribosomes. So don't get confused with rough endoplasmic reticulum and Golgi apparatus. The primary protein synthesis organelle is ribosome. Once ribosome makes protein, it could go to endoplasmic reticulum. So this is the basic cellular knowledge of structure. Now let's go into the organization of plasma membrane. So plasma membrane is essentially made up of lipid particles, and a lipid particle is made up of a globular head, and they have a tail, thin flagella-like tail. So this globular head, this is globular head, which is charged. Charge means they could be composed of positive and negative charges all over. So this is this globular head could be charged by the presence of positive or negative charges. So this is also called as polar, and they love water, so they're called as hydrophilic. So depending upon a particle or a substance, whether they like water or they don't like water. If they like water, it's called as hydrophilic. And if they don't like water, it is called as hydrophobic. So again, lipids are made up of globular head and a thin tail-like structure. The globular head is charged, it is polar, and it is hydrophilic. They like water. Now, as in contrast to the head, the tail has just the opposite properties. So they are non-polar and they don't like water. They dislike water. So they are non-polar, they are non-charged, and they dislike water. So what is interesting here, if you note, the interesting point is that this lipid molecule is demonstrating two different qualities, and which are very contrasting. Polar, non-polar, polar head, non-polar tail, hydrophilic head, hydrophobic tail. So this kind of property of lipid is called as, this is very important, this is called as amphipathic. Amphipathic means dual characteristic. Amphi means dual. Amphipathic means dual characteristics. Polar versus non-polar, hydrophilic versus hydrophobic. So what is the function of lipid? Lipids, they help in the formation of cell membrane. So let's see how they do. Just remember the structure. How they do is they arrange these lipid molecules, they arrange in two layers. So a plasma membrane is made up of two layers of lipids. And that's why the plasma membrane is called as bilipid layer. So let's say this is head, and these are the tails of lipids. Now in a membrane, there will be, as I mentioned to you, two layers of lipid. So let's say this is the plasma membrane, and let's consider that this is outside of the cell, and this is the inside of the cell. So outside and inside is differentiated by the plasma membrane, which is made up of two lipids layer. Hence, the plasma membrane is called as bilipid layers. Bilipid layers. This is the composition of a plasma membrane. So let's go to the last topic of. I mentioned that this chapter I'll be teaching in two small sessions. One would be cellular structure, lipids, and the third would be mitochondria, and the fourth will be enzymes. So mitochondria is the powerhouse. Why it is called as powerhouse? Because it generates ATP. So remember, when we eat, let's say if we eat bread, it is broken down into glucose. Glucose is further metabolized, and ultimately it reaches into mitochondria, and then there ATP is generated. You will be interested in it's very interesting to note that mitochondria contains its own DNA. And so it is assumed that the hypothesis behind is that mitochondria at certain point of evolution could be an independent bacteria that entered into alliance or symbiosis with another cell to give it the ability to generate ATP. So two different cellular bodies, they came together, and mitochondria became the part of the cell to generate ATP. And if you see these structures, this bluish structure, this is called as matrix. And these are folded structures which are called as cristae. Mitochondria also has its own ribosomes, and that helps in the generation of ATP. Okay, so the last subtopic in this part of the first part of this chapter is enzymes. So what are enzymes? Enzymes are, in 90% of the situations, enzymes are proteins, because as the technologies are coming around, it is noticed that certain RNAs can also work as enzymes, but enzymes are proteins. And what does these enzymes do? These enzymes increases the rate of reaction. So typically anything that enhances the rate of reaction is called as a catalyst. So enzymes are, because it supports life of a cell, it is called as a biocatalyst because and their maximum of them are proteins like metabolic enzymes. There are enzymes which are involved in protein synthesis. But as the technology is increasing, and we now know that some RNA also behaves, they have enzymatic property. So enzymes increases the rate of reaction. So that's it. So we finish first part of chapter 2. The second part of the chapter 2, we will go into cell division, and which will include mitosis and meiosis. I did this for not making this chapter bulky, but to make it as a too small fragment. All right, so I'll come back with the cell cycle. Thank you.